Thank you so much for having me here today. I'm really excited to talk to you about some of the research that I've been leading at Syntis Bio, particularly in the rare disease space. I'll spend a little bit of time talking to you about our Syntis Bio platform at the beginning of the talk today, because it's useful for understanding how we're using this platform to deliver this engineered enzyme. So yeah, let's get into it. So who is Syntis Bio? Syntis Bio is a company that was founded about 4 years ago out of research conducted in the labs of Giovanni Traverso and Bob Langer at MIT. We're a relatively small company, about 20 people, but growing. And the focus of our research is targeting the duodenum, so the upper part of the small intestine, to deliver oral therapies that otherwise would be poorly bioavailable or poorly efficacious. At the core of Syntis Bio is our platform technology called SYNT, which is shown in a really graphic way here, but I'll describe it to you. So SYNT describes a polymeric coating that assembles in the upper part of the small intestine, so the duodenum. This polymeric coating is comprised of a specific polymer called polydopamine. A specific polymer called polydopamine, and polydopamine is exactly what it sounds like. It's polymerized dopamine monomer. So dopamine, just like you know that you've heard of as a signaling molecule in the body, those can actually polymerize to form a really thick polymer coating that can line the intestine and will be excreted with normal mucosal turnover over the course of like 24 hours. This chemistry was actually biomimetic. It was inspired by the adhesive proteins that the sea organism mussels use to adhere to various like wet and slippery surfaces. So we've taken advantage of this natural chemistry in order to improve drug delivery. And so how this works is for across all of our programs in our SYNT platform, we deliver dopamine monomers with a very small amount of peroxide. This peroxide catalyzes the reaction of dopamine to this polymeric coating polydopamine when it encounters very specific enzymes that are expressed preferentially in the duodenum. And these are various peroxidases that are there to aid in normal digestive processes. We, in particular, are typically referencing catalase expression. So catalase is fairly abundant in the upper part of the GI tract relative to further down, and especially higher than that, which you would see in the stomach. And so when dopamine monomers, along with the peroxide that we dose them with, encounter these endogenously expressed enzymes, you get that really quick in situ polymerization in the duodenum. So think about it as a coating in the duodenum that we can then deliver effective molecules for across several different programs. And today I'll obviously be talking to you about the rare disease programs. So both Homocystinuria and MSUD is another one that we're working on. So when we get this polymeric coating to form in the intestine, we can do a few different really unique things with it. And we can do this by engineering the polymer to form in different ways. So I'm gonna be primarily focused on what you're seeing here as retain for the HCU program, but I'm gonna speak to you just briefly about our redirect program because this program is in phase 1 clinical trials and it's validating our platform in terms of safety. So this coating can form in the duodenum and shunt nutrient absorption from the proximal to the distal bowel. And what we see when that occurs for this redirect program, so not applicable to the HCU program, but again, validating the safety for the HCU program, we can invoke a metabolic rewiring that leads to weight loss. And so this is for treatment of type 2 diabetes and obesity. Retain, this is for the HCU program. We can engineer the polymeric coating so that we can deliver enzymes that act on methionine in the local lumen of the small intestine. These enzymes would otherwise get cleared rapidly by mucosal turnover and normal transit through the GI tract, but we can use that polymer to really keep those enzymes at the site of methionine absorption so that the methionine doesn't have the chance to be absorbed. So unlike what Travere is doing, where they're targeting the action of like CBS directly, we're focusing on limiting the amount of methionine absorption upstream of that. And then the absorb program, This is a different program in which we try to localize things to the small intestine to increase bioavailability, but it's not relevant for this. Okay, so here are the two programs that we're working on in the rare disease space. So SYNT 212 refers to our Homocystinuria program. Again, the target is an engineered enzyme called methionine gamma-lyase. And I think that some of you may have heard about this before because it's been presented in work spearheaded largely by Kadexis and Kristen Skvorak, as well as Warren Kruger, who is on the original manuscript for some of the mouse studies. But what it basically shows, and I'll show you some of that data today, is that you can use this engineered enzyme that was isolated from bacteria and engineered to be highly resistant to intestinal proteases and stomach acid to degrade methionine in the lumen of the small intestine. So again, we're combining this enzyme with our SYNT technology so that it's an oral therapy that you can take once daily rather than at each meal, and it will lead to a reduction in overall methionine and downstream homocysteine. The stage that this is at currently is that we have validated it preclinically, and we're gearing up for IND-enabling studies within the next year. SYNT 213, it isn't as relevant for the HCU audience, but I just wanted to bring it up because it is also validating that we can do this across enzyme programs. So this is an enzyme that's been engineered to treat maple syrup urine disease, another inborn error of metabolism. And this is an engineered LDC enzyme that, again, Kristen was working on. Okay, so here are some of the early data for the enzyme alone on the top half of this slide. So if you look at the top left, what you're seeing is the plasma total homocysteine in rodent models after daily administration of the enzyme alone. So this is without the SYNT platform, after 7 and 14 days of treatment relative to the vehicle. And this is when exposed to a daily whey challenge and on a low MET diet. And what you can see is that with treatment, you don't see an increase in the total homocysteine, where of course with the vehicle, you see a 39% increase over the course of that 2 weeks. So this was in the mouse model. This is a mouse model that does have homocysteine in a healthy human primate model, so a larger animal species. What Kristen's team saw is that there's dose response activity in terms of increasing doses of the MGL enzyme lead to decreased methionine plasma levels, and this is after a single treatment at each of these doses, and this is in a healthy non-human primate. So what we saw is when we tried to start using this enzyme at Syntis in our own hands is that it worked just the same way. However, after a few hours, we saw clearance of the enzyme just with normal GI tract motility, and the enzyme no longer had efficacy after a few hours if re-challenged subsequently, and this was in a rat model that we were trying to do this. And if you look at the bottom here, I have a result here that summarizes what we saw, but I'd like to get a little bit more into the details of the data here, which is over the course of 8 hours, we administered MGL. This is an MGL, the same engineered MGL that we have performed a various coding strategy with so that it will be an extended release in the GI tract along with SYNT, and so that it can better handle some of the stresses that are involved with this oxidative reaction when SYNT forms. Over the course of the 3 challenges over the 8 hours in a healthy rat model, we saw a 26% reduction in plasma homocysteine relative to the MGL alone, which loses efficacy at that 3rd challenge. So this is part of our initial proof of concept to show that SYNT does work to retain the enzyme in the lumen of the small intestine, so this was exciting. We've since done this in other sorts of ways and different time frames, and excitingly, we do see that SYNT seems to have activity of MGL or sustains activity of MGL over the course of 12 hours. So this is really exciting to us to enable an oral daily treatment as opposed to having to take the enzyme once for each meal. For SYNT 213, we have yet to combine it with SYNT. It's something that I'm currently working on. I know this isn't as relevant to this audience, but I just want to show that the same enzyme leads to a lowering in the rodent model with the engineered enzyme, as well as the non-human primate. We get dose response activity across the 2. And we're now working to combine this with SYNT as well. Okay, so I would like to thank everyone who's supported this project, particularly the HCU Network America as the host, and your support, and the NIH for our grants, and then for Syntis Bio, the whole team, and then especially Warren Kruger at Fox Chase Cancer Center and Kristen at Codexis for the foundational work that has been vital for us implementing SYNT with the various engineered enzymes. And with that, I will take any questions on the technology. Yeah.
Present within the GI tract, or is there a way to throttle it, if you will?
Yeah, great question. No, it's not. So like, for example, for one challenge, we see about a 30% reduction in the methionine, and we think we can engineer this to a bit with the ratio of like the enzyme to the SYNT platform as well.
I see.
Yeah.
Thank you.
Yep.
That's exciting.
Yeah, thanks.
Hi, Ashley. Very nice talk. So I was wondering with the new studies that you've done with the SYNT technology with the HCU molecule, I know it's naturally cleared after 24 hours, so there is some residence time there, but do you think it's active along the entire digestive tract? Is it coating that whole organ, or is it really only working in the duodenum?
Yeah, that's a great question. So the initial formation of the polymer is primarily the duodenum. That sloughs off with normal mucosal turnover. So the majority of the time it is in the duodenum, and then it's more of a swift, like just the same time as mucosal turnover further down in the GI tract. And we've seen this. So we've done like some pill cam studies in a beagle model. And we've seen that, it's really in the duodenum and really concentrated there for quite some time, and then it's more rapidly cleared. But I suppose if the challenge was administered at a time in which it was further down in the GI tract, there would still be some efficacy there just from the enzyme being there.
Okay, so just follow-up question, sorry. You do believe that it is active throughout that 24 hours? Have you ever
Yes.
looked at the output, for lack of a better word, and testing if there's still activity and what's being excreted?
Oh, no, we haven't. That would be fun to do though. Yeah, that's actually a really good idea. The furthest we've tested right now in the rat model is 12 hours, and we really want to do these chronic dosing studies soon. We're trying to figure out, so the interesting part about the technology is that it has really good efficacy as a solid dosage form, and we're developing these like microparticulate formulations, and we're having trouble dosing them to like small animals just because it's hard to gavage those to mice. So we've been like really limited to rats and pig models right now. But I suppose a pig model would be more like relevant for human intestinal transit times. And that's where I think it becomes really interesting.
Is there any type of work being done to ensure that you're not preventing the absorption of other like vital nutrients in addition to the methionine?
Yeah, that's a really good question. So 2 points there. So because the SYNT platform has that barrier function that we see for our obesity program, we have tuned the formulation of SYNT to not be as dense for this program. So you're not seeing any, so the SYNT polymer coating does not restrict any methionine absorption. Actually, it's all the action of the enzyme. We've done those tests. The only thing that I think we need to look into a bit more, but I think there's published data out there, is like the specificity of the enzyme against other amino acids. Yeah.
I have a question.
Yeah.
So have you tried this formulation in any of the mouse model, like CBS knockout or transgenic? And if so, can you give us a sense of how much the homocysteine changes?
Yeah, so we haven't been able to dose the one where it's coupled with SYNT in those models just because of that, like the particle formation or the particles that we're trying to deliver are difficult via gavage, but it's something that like every day I'm testing.
Okay.
So like to try to get the syringe to work.
Okay.
So
Wonderful.
it's like very much our hope that we'll be doing that soon. Yeah.
Excellent. Great. Thank you so much.
Thank you.